Biotechnology. We'll be covering these topics. We'll start with the tools of the microbiologist, then food preservation, selective breeding, biotechnology in agriculture, and then biotechnology as it applies to human health. Tools of the microbiologist. When we use water delivery systems and filtration to remove microorganisms from our drinking water, we are using technology to keep ourselves free of infectious disease. When we engineer sanitation systems to keep our food and water clean, we are using technology to control the growth of microorganisms in our environment. When we use heat, detergents, soaps, and alcohols to keep our bodies and homes clean, we're using technology and our understanding of microbiology to stay healthy. Microorganisms can be used as tools to cure diseases and improve the quality of our lives. They use fungi, bacteria, and viruses as model organisms to understand the characteristics of living things. They use Chromista and Protozoa as model organisms to understand principles of ecology. Genetic engineers use bacteria and viruses to modify genes and to move genes from one organism to another. Microbes are cultured when they are grown in the lab so that they can be studied or used as tools. While individual microbes are not visible, pure colonies of one species can be grown under lab conditions and can be examined in different ways. Colonies of bacteria can be seen when they are grown on nutrient agar. Agar is a solid gel derived from seaweed, similar to gelatin. The nutrients added to the agar include amino acids, salt, and extracts of yeast or beef that provide other biomolecules. A petri dish with nutrient agar ready to receive a culture. To grow bacteria in a controlled way, sterile petri dishes, nutrient agar, and deionized water are required. It is important to use sterile technique so that only organisms of interest are grown. If researchers touch the agar with their hands or non-sterilized tools, breathe, or even speak near the agar, other bacteria could be introduced, resulting in a contaminated culture. These scientists are using sterile technique to grow insect larvae. And you can see we've got gloves on here, got protective suits, hair is bundled up, and it doesn't look like they're talking to each other either. To grow a colony of one type of bacteria, it is added to an agar plate at a low concentration so that each cell has room to grow. Each individual cell will replicate many times and give rise to a visible colony of genetically identical cells called clones. The scientist is producing the COVID-19 vaccine using cultured bacterial cells. Each dot on the plate here, there's a whole bunch of them, is a clone. The petri dish has cultures of 6 types of bacteria that can cause urinary tract infections. In areas where low concentration of cells were added, individual colonies can be seen. And it's a little tricky to see, but the one in there, that's the little blue dot. And over here can be that little blue dot. It can also be the little white one. These are clones that arose from a single bacterial cell. This culture of bacteria shows the effect of antibiotics. Each white disc over here, have several of them, is releasing an antibiotic. The beige bacteria, that's this color here, kind of like that light brownish gray, that's where the bacteria are growing everywhere except within these circles around the antibiotics. Nothing is growing in there. Visit the links below to learn how to set up nutrient agar plates and culture a variety of microbes in your environment. If the materials listed below are not available, effective substitutions, such as those listed on the next slide, will also yield bacterial colonies. So on the left side here, we have lab materials, petri dishes, agar, nutrients that are found in agar, and a source of bacteria. If you don't have any of that, here are some substitutes. You can use plastic food containers or lids that are sterilized by boiling or with rubbing alcohol. And instead of agar, you can have pure seaweed gelatin boiled for 10 minutes and made at a 3 times concentration. Nutrients, a low sodium bouillon cube added to the gelatin solution, a bit of sugar. And finally, if you need a source of bacteria, just cough towards the plate. Touch it with your hand. Swab doorknobs, countertops, pencils, water fountains, etc. Bacteria is everywhere. Here we have students streaking plates, over here, with bacteria found on their hands. And again, that is on everybody's hands. Every environment and every individual has a unique set of bacteria. When cultured, each species of bacteria has a characteristic colony appearance in terms of color, shape, and size. And it looks like a person took their entire hand, put it on this plate, and then this was cultured, and you can see the different types of bacteria all over that person's hand. And again, everybody has something like that. That's not unusual. Nutrient agar will also support the growth of fungi. Colonies of fungal hyphae have a fuzzy appearance. It's over here. The gray and white colonies are probably fungi. This can be verified by looking at samples under the microscope and looking for hyphae and sporangia. Mold and fungus are both members of the kingdom fungi. Molds are multicellular, while fungi can be unicellular, that's one cell, or multicellular. Molds and fungi grow as networks of hyphae. Hyphae are tubular structures that release digestive enzymes and absorb digested nutrients and water. Mold spores are the reproductive cells released from sporangia, the modified hyphae that produces spores. Culturing a specific species of fungus or mold in the laboratory requires a source of the species and technical expertise. But it is still possible for students to observe fungi and mold in our environment. Mold and fungal spores are present on most organic surfaces and will grow under warm, humid conditions. Bread or fruit that has been allowed to rot in a humid place will usually grow mold. If you make a slide from a sample of mold or fungus and view it under a microscope, you will see hyphae, that's these strings here, and sporangia over here. Rhizopus, hyphae, and sporangia are visible. Rhizopus is also called black bread mold and is very common. Culturing particular species of Protozoa and Chromista requires knowledge of the organism and specialized growing conditions. To view a variety of unicellular eukaryotes, however, you may view samples of water from a lake or the ocean under a microscope. For a beautiful slideshow of Protozoa and Chromista under the microscope, follow the link below. Viruses are difficult to culture because they cannot grow independently of host cells. Culturing viruses requires growing appropriate host cells and then infecting them with isolated virus particles. Virus particles are too small to be viewed with light microscopes. Infected host cells often have disrupted cell structures, so microbiologists can observe the effect of viruses, not the viral particles themselves. Virus-infected bacterial cells are lysed. That means their cell membrane is broken down when viral particles are released. The blank spots in the bacterial lawn are spots where a virus is replicating.